Welcome to part 2 of the nervous system lectures here, where inside the cell we've pumped a bunch of K+, and outside the cell we have even more Na+. So we have this imbalance, but specifically there's certain ions outside and certain ions inside. Now we call this imbalance, where it's essentially negative 70 millivolts inside, we call that a resting potential. This is kind of the norm for any type of neuron cell. But we also have these gated ion channels, which allow for, if triggered, the gates to open, and they allow specific ions, whether it's sodium or potassium, to pass through. So if I open up a series of sodium channels, that's going to depolarize things. It's essentially going to make me go from negative 70 millivolts and go up towards 0, right? So that's depolarization. This is what happens if I open up Na+ channels. If I open up K+ channels, it's actually going to make me move to be more negative, because if K+ leaves, because there's not much K+ outside, so the K+ says more in, less out, I'm going to move where there's less. And so the positive charges leave the cell, making it more negative. Whereas with sodium, they're packed on the outside, they want in, they're also positively charged. So as sodiums move in, it makes the insides less negative. So these will be critical, because as our cells, as these sensory nervous cells as these sensory neurons receive stimuli, they are ultimately going to, in most cases, open up sodium channels, and enough of those sodium channels, if they open, will allow enough Na+ in to cause an action potential. So this is kind of showing things here. This is our resting potential. Then some stimulus occurs that is going to open up these sodium channels, and it's going to depolarize the neuron. If it depolarizes it enough to reach this threshold, which is usually negative 50 to negative 55 millivolts, somewhere in there, if it reaches the threshold, then it completely depolarizes. All these sodium channels open. You have where there's a massive shift to where the cell actually becomes slightly positive for a little bit instead of negative inside. And then after you're done with this depolarization phase, you close these sodium channels. So right now this is Na+ open. And then over here, we're going to say Na+ are going to close, and we're going to open up these K+ channels, which we said make us more negative. So they open, and then the charge starts to dip back down. You're also going to use the sodium-potassium pump at the same time that's going to be going on to try to make sure, because before you fire again, you have to get back to this original state, this original resting state. We call this lapse the refractory period, where even if we have these shifts, if we don't have an overabundance of Na+ outside the cell and an overabundance of K+ inside the cell, it won't work. You know, the sodium channels only work effectively if there's more sodium outside than inside. The potassium channels work effectively if there's more potassium inside than outside. And so during this period here, you'll also have the Na+ and K+ pumps pumping the K+ in and pumping the Na+ out to get us back to our normal level, at which point you could have another action potential occur. Now, I'm not going to go through this slide. This is just kind of outlining the steps I just mentioned. So I'm putting it here, so if you want, you can pause and you can look over it. It's just summarizing what I've said. But this just kind of maps out that cycle we saw on the previous page. Okay, action potentials. Once they occur, they self-propagate, meaning it's going to go from the dendrites down to the axon terminals all by itself. We don't have to keep making it happen. By depolarizing one section, it triggers the next section to depolarize as it moves along the neuron. It also, this action potential, is going to trigger the synaptic vesicles that we'll talk about again in a second to be released to then trigger an action potential on the next neuron potentially. So this will kind of get itself going. It's going to be one directional in almost all animals. We've said there are some kind of oddballs like jellyfish that are very primitive that still have two directional stuff. But in general, it's always going to go from dendrite to axon. And we can adjust the speed of this. If you get a large axon, that makes it easier for this charge to move along the axon itself. And so in ideal conditions, you can get about like 100 meters per second is the speed you're looking at, which seems really, really fast. But like in a 6-foot tall person, if you stub your toe, you are going 2 meters down and 2 meters back up. So depending, or I should say 2 meters up perhaps, you'd say to the brain, and 2 meters back down. So that's a total of 4 meters. So there is going to be a fraction of a second before it receives things. And if your axons aren't particularly large in diameter, then you're going to get slower speeds. And so that means this process can take even longer. And so if you're talking about 50 or 10 meters per second, you can now have where it might be almost half a second before there's some type of response. Because if you'll go 10 meters per second, that seems crazy. But we already said to go up to the brain and back down, that's 4 meters. Well, 10 meters per second, 4 is almost half that. So that's almost half a second it would take for you to stub your toe, receive that in your brain, and immediately, we're assuming, send a response back. It'd take half a second just to do that. And so you can start to realize that the nervous system has to be very fast for us to appear to be, interpreting stuff in real time and not lagged. The other way you can speed this up is use saltatory conduction. You'll see a lot of axons will have this layer of myelin called the myelin sheath. And so it's just insulated. And what this does is it allows the charge, the action potential, to kind of jump from these, they call them nodes of Ranvier, but these kind of blank spots. So it can hop. And by hopping, it actually speeds up the rate at which it can go. So if you use saltatory conduction, you can get on ideal conditions up to about 150 meters per second, which at that point you're talking about a very, very, very tiny amount of lag between something happening and you responding to it. So that's going to be very useful, especially for big organisms that have to have things travel long distances, like a giraffe or maybe like a blue whale, where you're talking what might be a 100-foot-long organism. If it's going to detect you grabbing its tail, that's got to travel 100 feet up to the brain and 100 feet back. That's 2/3 of a football field. That's a large distance. And so 150 meters per second can kind of help that happen better. So it's not going to be like 2 seconds later that it starts to shift its tail because you touched it or stabbed it or something. Now, the action potential, just to make sure we're clear, is going to come down the presynaptic cell. That's going to be this guy right here. So this one's presynaptic. And as it rolls along, it's eventually going to hit these calcium channels and allow calcium to go inside. So we've got calcium 2+, which can then go into the cell. And when calcium 2+ gets inside, it can bind to these vesicles and trigger them to go here. Well, in this case, I'm going to draw it shorter here. Just go boom. And go through exocytosis to release the neurotransmitter into the cleft, into this gap. They can then diffuse over, they can bind to the neurotransmitter receptors that'll be over here, and then that can trigger the sodium channels to open, which can trigger the action potential to start again. So this process will be dependent upon this action potential because it does trigger the whole release of neurotransmitters from these synaptic vesicles, which then triggers the opening of these sodium channels to start the action potential on the next neuron. And as we kind of get close to wrapping up here, I want you to realize that some of our particular neurotransmitters will depolarize things. So they're what we call excitatory. You'll also have some of them will be inhibitory, which means they'll hyperpolarize. They're going to make it more negative. So in some cases, you're going to make this more positive. In other words, you're going to make it move towards an action potential. And in some cases, you're going to make it less positive, more negative, which means you're going to move away from an action potential. You're going to make it less likely that we have an action potential. So don't assume all stimuli say go, 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 do something, do something. There are stimuli and neurotransmitters that can be released that say, let's not do something. Let's make it where we don't fire rather than just making it where we're more likely to fire. And then the last one for this section is just neurotransmitters themselves. The most common guy I'm going to bring up is acetylcholine, because acetylcholine is the one that's used throughout our peripheral nervous system, your arms, legs, the stuff that connects to your muscles, most of your glands. And so this is one of the most common neurotransmitters that there is. But inside the brain, I want you to realize there's lots of different neurotransmitters, which is what fits in with that idea we just mentioned, where some can make you say let's go and some can say let's not go. Let's be moving towards threshold, let's move away from threshold. And so some of the examples are there's groups. The biogenic amines have norepinephrine, which you guys may have heard of. Dopamine, that one's the one that's commonly indicated with a lot of brain chemistry issues where you might take dopamine for it. Serotonin has been linked to at least some role likely in depression, whether it's the cause or more symptomatic, but this is what a lot of stuff will go after that you take that are antidepressants. So these are all ones that are frequently used in the brain. Endorphins, this is the one that runners, sex, things like that release that tends to make you feel a little bit high. It's like a opiate. It dulls pain, makes you feel kind of euphoric. And so that's a neuropeptide. So it's kind of like a really mini protein, if you will. And there's a lot more. I'm just giving you some basic stuff. And I figured I'd throw nitric oxide on there because we mentioned it before in chapter 45. So we did say it can be used as a neurotransmitter. So in small quantities, it can be used as a neurotransmitter. So within the brain especially, you'll see that there will be a lot of different neurotransmitters that can be released, that can be involved based upon what's going on. And that helps give us the tremendous complexity we have within our brains to do all the amazing tasks we can do, such as you listening to this, interpreting and storing this information, and potentially being able to explain it to me at a later date. It's fairly impressive, however you want to look at it.